with that, I'd like to introduce our next talk. First of all, I'll introduce Chad Grueter. Chad is the Vice Chair of Research and Associate Professor of Internal Medicine at the University of Iowa. He studies how genes like MED13 and MED13L, a key part of the mediator kinase complex, control heart function and whole body metabolism. His lab uses mouse models to uncover how disruptions in biological systems contribute to heart disease, with broader implications for understanding how MED13L mutations might also impact neurodevelopment. Dr. Grueter's findings help connect basic genetic mechanisms to real-world health outcomes across the body. Chad was here a few hours ago. Unfortunately, he had a medical emergency, so he had to run out. He's fine. In his absence, we have Kayla Henry, who is a graduate student in the Grueter lab, who will present his materials. So, welcome.
Okay, so like it was said, I am sadly not Chad. I am instead his grad student doing my PhD in genetics. But today I will be talking about his title, Mediator Complex in the Heart and Brain. So a little bit of an overview of what I'll be talking about. I'll start off with a little bit about gene expression, moving into the mediator complex and the kinase submodule, specifically MED13-like, and then some future directions with MED13-like tissue-specific studies. So if we think about the genome itself, about 2% of the human genome actually encodes for actual genes. And then if we compare across humans themselves, we're about 99.9% similar with humans, 85% similar with mice, and 32% similar with yeast. So that really shows that there's a high conservation between our genes across different species. And so understanding how we're using the different pieces of our genome to really make us all different people is really crucial to understand. So if we take a look at what the central dogma of biology is, we can think of our DNA, which is our genome that we've kind of been talking about. It's full of those genes. And then from there, it undergoes a thing called transcription to get turned into RNA. And so those are the genes that we're actively expressing. From there, that RNA gets translated into the actual protein that each of the cells are using. So understanding how we know exactly what genes we want to express off of this DNA is crucial for understanding why we have different cell types. So again, we can think about this, that every cell in our body has the exact same DNA, but they're all using it differently. So that's how we know that we have different muscle cells versus neurons, right? They're expressing different genes. And so in order to know what genes to be expressed, we have to have different spatial and temporal controls, understanding what genes need to be turned on versus off. So those consider or control whether a cell has a state or its fate for knowing if it's a cardiac cell or a neuron, liver cell, etc. The mediator complex itself is crucial for understanding this. So as I'm sure a lot of you guys are aware, we'll go over a brief little refresher here, but the mediator complex itself has a core 26 subunits. It then has this reversibly dissociating kinase submodule that has cyclin-dependent kinase 8, cyclin C, which is going to be crucial, and remember that for Randy's talk later, MED12, and then MED13 or MED13-like. And the mediator complex is really critical in helping to get RNA polymerase II, which is kind of the workhorse for driving actual gene expression, in the right spots. So the mediator complex really controls where these genes, or where to find these genes within our genome. If we take that one step further, we can look at mediator transcriptional control, in which case things like transcription factors or these different promoter regions are really important for getting the mediator to the right spot, where MED13L really plays a big role in making sure that the mediator complex is formed properly and has that bridge between the kinase submodule and our actual mediator complex. So how exactly is the mediator guiding this transcriptional control outside of just having these binding partners like transcription factors or these different regions that we call promoters. So it integrates specifically with cell signals. So when we say that it's regulating gene expression, we're kind of talking about that it's controlling the speed and the amount of each of these genes that we're creating into these proteins. So how quickly are we making them, or are we making a huge amount compared to just one copy here or there? By gene expression, we're using these different informational tools from the gene to express the protein. So when we combine that together, we're talking about regulated gene expression, so how the cell controls the speed or amount of the gene being expressed as the protein. So if we take a look here at this little schematic, it's showing that this kinase submodule is important for understanding the different environmental factors or what's going on within the cell or outside of the cell to really change what genes are getting RNA polymerase II to bind to and actively transcribe and start this transcription. So it plays a really big role in integrating what genes we express and how our cells are responding to our environment and what states or fates they want to be in. So next we can think about how we're going to study this. So our lab specifically does different transgenic mouse models. And so we already talked about that mice have a pretty high similarity to humans. And using mice, we can delete MED13-like in specific tissues for a temporally controlled manner to make sure that we're really figuring out what MED13-like is doing. So when we think about that, with our mice, we can add these fun things called little LOX-P sites outside of regions that we want to delete in the mouse. And so using that, we can add different things called Cree. And so Cree will come in and take our little LOX-P sites, they'll overlap with each other and excise that chunk. So this little blue part would then be gone from our mice, and we can now have this knockout model to really test what's going on without MED13-like. So this really helps model the development of the human disease in this controlled environment because we can control what different cell signals that they're getting and what maybe these mice are responding to. And this would allow us to replicate specific characteristics, symptoms, or the pathology of these diseases, and then test potential therapeutic approaches once we figure out kind of the mechanism that's going on. So what we've learned so far from our MED13-like mice is that we know that MED13-like is expressed in both the heart and the brain. Our mice so far have shown that if you knock out both copies of MED13-like, it is lethal. However, just the heterozygous, or they have one correct copy of MED13-like, or they have one knockout version of it, they do have normal cardiac function. If we look at our MED13L in different neurons, here we can see that this is what our control or our normal neuron should look like. If we look at the knockdown of MED13-like, you can see that there is a decreased neuronal branching here. So we're getting this abnormal neuron structure. We can also look at the RNA sequencing data. So if we think back to what our central dogma is, right? We have our genome that then gets transcribed into our RNA. So that first step, really looking at what is actually happening at a transcriptional level. We can look at what was being transcribed, specifically in a region called the hippocampus in the brain. And from there, we really saw that there was this downregulation of different transcriptional properties and different MAPK kinases and kinase activity in general to help get an idea of what's being impacted by MED13-like. And then finally, and we'll hear about this later, MED13-like have craniofacial and behavioral differences, so we can really start to target those and figure out what's going on mechanistically on that front. In the future, our lab is going to continue our different phenotyping studies. We're specifically going to start targeting tissue-specific studies with MED13-like. So we're going to isolate MED13-like to the neural crest, which is a really important player in development. And so really driving these different cell state switches as you're developing, so telling the cells that they should become cardiac cells or neurons. So really figuring out what's going on there, specifically by doing some RNA sequencing. And then that'll really help the mechanistic understanding of MED13-like function. We're going to be testing some different therapeutics and then also working in collaboration with Dr. Brad Grueter at Vanderbilt to investigate the neuronal activity with electrophysiology. So with that, I'll thank the different Grueter lab members, our different funding sources, and of course the foundation for not only giving us the opportunity to be here and meet all of you guys, but the funding to do this research, which is incredibly important. With that, I will pass it on.